Today, in the second out of the third lecture on the cell membrane, what we're going to learn about is the process of diffusion and osmosis. So, diffusion is a tendency for molecules to spread out evenly to fill the available space. What does that definition mean? Okay, well, for example, let's say I have a glass of water, and I put a drop of food color and a food dye at the top of the glass of water. Okay, initially there's a lot of, let's say, blue food dye at the top of the glass of water, but none at the bottom. Okay, but then I just let that glass of water sit for a few minutes, and what happens is that dye starts to spread, like the dye molecules spread through the water molecules, to in the end make one all pale blue colored glass of water. Okay, so what's happening is the dye molecules are diffusing through the water molecules. Or, let's say for some reason, I'm in a classroom, and at the front of the class I decide I just have to put on some cologne. And I spray on some cologne. Well, initially, the students near the front of the class smell that cologne, but the ones sitting near the back don't smell it. But give it just a few minutes, what will happen is the cologne molecules, the odorant molecules, start to spread or diffuse through the air molecules, and the students in the back of the classroom will also be able to smell it. So that's what diffusion is. Things are spreading out evenly to fill the available space. Now, diffusion can also happen across a cell membrane. So let's say we have a bunch of this blue dye molecules outside the cell, like we can see here, okay? And we don't have any at the beginning of that blue dye inside the cell. What's going to happen, let's say this membrane is permeable to this dye, so the dye molecules can pass through the membrane. What's going to happen is those dye molecules are going to start diffusing from over here to over here. And they're going to keep doing that until there's an equal concentration of dye molecules on both sides of the membrane. Okay, so that's what diffusion is. Now, of note, the net direction of diffusion is always from an area of high concentration, there's a lot of dye molecules here, to an area of low concentration, there's not many dye molecules here. So you can see here, high concentration to low concentration. That's the net direction of diffusion. And that continues until you reach equilibrium. And equilibrium is when there's an equal concentration of dye on both sides of that membrane. Now, of note, if you were paying attention, I said the net direction of diffusion is from an area of high concentration to an area of low concentration. Now, I was very specific when I said net direction of diffusion. The reason is because it's not like all of the dye molecules are trying to diffuse down. It's not like they're all trying to go from here to here. Because if they were all trying to go from the top to the bottom, at the end, they'd all be on the bottom. There wouldn't be any at the top. Okay? Or in my glass of water, when I had a dye at the top, if it was all trying to go to the bottom, at the end, you'd only see blue dye at the bottom of the glass. It would be clear water at the top of the glass, but that's not what happens. When I spray the cologne, if the cologne molecules were all trying to go to the back of the room, at the end, you could only smell at the back of the room, you wouldn't be able to smell at the front of the room. That's not what happens. What happens is it spreads out evenly to fill the entire available space. So that's what diffusion is. So they're not all trying to go in one particular direction. What's happening is each dye molecule is moving randomly, okay? It's completely random which way it's moving. But since there's more of them here and less than them here, overall the net direction of diffusion is from high concentration to low concentration. An individual dye molecule could be going from the top to the bottom, or it could be going from the bottom to the top. Okay, but overall, since there's more here and less here, the net direction is from high concentration to low concentration. So when you reach equilibrium, the other thing that means is these molecules have not stopped moving. They're still moving. But since there's equal concentrations on both sides, anytime one is moving from the top to the bottom, one is also moving from the bottom to the top. So the net direction of diffusion is from an area of high concentration to an area of low concentration. That's the net direction of diffusion. Now, there's two other ways of saying that mean the same thing. You got to know all the different phrases for how to say that. So the net direction of diffusion is from an area of high concentration to an area of low concentration. Or you can say something goes down its concentration gradient. That means from high concentration to low concentration. or you can say something goes with its concentration gradient. That means from high concentration to low concentration. So high to low concentration, down the concentration gradient, with the concentration gradient, that all means the same thing. Now the other thing to note is the cell doesn't have to do any work to make diffusion happen. Diffusion happens all on its own. When I put the dye in that glass of water, I didn't mix the dye, I didn't stir it, I just let it sit there and it diffused all on its own. When I put on some cologne, I didn't put a fan to blow it towards the back of the room, it went towards the back of the room all on its own. Okay, so no work has to be done for diffusion to happen. Now, an analogy to help you try to keep this in mind, it's kind of like the example of if you have water at the top of the hill, water will very easily roll down the hill all on its own. Okay, you don't have to push water down the hill. Water will roll down the hill all on its own. Similarly, things diffuse down their concentration gradient all on their own. You don't do any work to make it happen. So, since it doesn't require the cell to spend any energy to make it happen, diffusion is called passive transport. Passive transport means it doesn't require any energy to make it happen. The other type of transport would be called active transport, and that does require energy to make it happen, and we're going to talk about that later. Okay, but that'd be the analogy for active transport would be water going up a hill. If you want water to go up a hill, it's not going to go up a hill on its own. Okay, you have to spend energy, like electricity, to pump water up that hill. Okay, but that would be active transport. Right now we're talking about passive transport. It doesn't require the cell to spend any energy because things are going down their concentration gradient from high concentration to low concentration. That's an analogy for that is water rolling down a hill. Now, very related to diffusion is a term called osmosis. So all osmosis is the diffusion of water. So since water is so special, it's absolutely required for life, we give the diffusion of water a special name. We call it osmosis. Now, I'm going to read you the definition of osmosis. It might probably sound a little confusing when I first read it to you, and then I'm going to explain it with an example in the next figure. So water diffuses across a membrane from a region of lower solute concentration to a region of higher solute concentration until the solute concentration is equal on both sides of the membrane. Okay, so what was that actually saying? So here's an example. Let's say we set up a little experiment where we have this beaker here, and in the middle of the beaker we have a semi-permeable membrane. What that means is some things can cross this membrane and other things cannot. Okay, and then in the two different sides of the beaker, what we're going to add is let's say, sugar water. So water that has sugar dissolved in it. But on the two different sides of the beaker, we're adding two different concentrations of sugar water. So on this side of the beaker, we add a dilute sugar water solution. So there's not much sugar dissolved in this water. On this side of the beaker, we add a concentrated sugar water solution. In other words, there's a lot of sugar molecules in this water over here. Okay, so it's like, this is like really sweet tasting sugar water, and this is like dilute tasting sugar water. So there's a concentration difference of the sugar molecules on the two sides of the membrane. And sugar, just like everything else, wants to diffuse down its concentration gradient from an area of high concentration, what you have over here for the sugar, to an area of low concentration. So the sugar wants to go from over here to over here, but it can't, because it can't fit through that membrane. Okay, the membrane is not permeable to sugar. Sugar cannot pass through it. So although sugar wants to go from here to here, it can't, because the membrane won't let it pass through. Now, what can easily cross through this membrane from one side to the other is water. Water can easily cross from this side to this side. And water, just like everything else, wants to go from an area of high water concentration to an area of low water concentration. Okay, well, in this beaker, it turns out I have more water over here and less water over here. Now you can look at that and say, no, that's not true. Look, it's the exact same amount of water. They're filled up to the same. Okay, so to explain that one, Let's say that I have 2 glasses, and into one glass I add like 3 tablespoons of sand. In the other glass I don't add any sand. And then I fill both glasses all the way up to the rim, the top of the glass, with water. Now, everybody that's listening right now would agree that the sand-water glass has less water in it than the water-water glass, because the sand is taking up space. So that glass has less water inside it because the sand is taking up some of the space. Now let's say I do the exact same thing, except instead of adding 3 tablespoons of sand, I add 3 tablespoons of sugar. And then I fill it all the way up to the top. Now that sugar is going to dissolve. It's going to like turn invisible. You won't be able to see it, but it's still there, which means it's still taking up space just like the sand was taking up space. So the sugar water glass has less water in it than the glass that has just water, even though they look exactly the same. So this is kind of like the sand water glass, and this is like the water glass. This one has less water in it than this one because there's more sugar molecules taking up space. So even though they're both filled to the same level, because there's a lot of sugar here, it's taking up space, so that means there's less water here. Okay, so water, just like everything else, wants to diffuse from an area of high concentration to low concentration. Okay, well there's an area of high concentration of water here, a lot of water here. Area of low concentration of water here, not a lot of water here. So what does the water do? It diffuses from an area of high concentration to low concentration. And it continues to do that until the water concentration is equal on both sides of this membrane. Okay, and that's what you have over here. Okay, so that's osmosis. Osmosis is diffusion of water. Water goes from an area of high water concentration to an area of low water concentration until the water concentration is equal on both sides. That's what osmosis is. One of the reasons osmosis sometimes is confusing is because how we refer to it. Because with osmosis, we don't normally refer to the thing that's moving. We don't normally talk about the water molecule. Instead, what we normally talk about is the thing that's not moving, which is the solute, the stuff dissolved in the water. In this example, sugar molecules. Instead of saying water diffuses from an area of high water concentration to an area of low water concentration, instead what we say is the net direction of osmosis is from an area of low solute concentration to an area of high solute concentration until the solute concentration is equal on both sides. That's the net direction of osmosis. Okay, so the direction of osmosis is from a low solute concentration, which means high water concentration, to a high solute concentration, which means low water concentration, until the water concentration is equal on both sides. Yeah, so one more time. The net direction of osmosis is from an area of low solute concentration to an area of high solute concentration until the solute concentration is equal on both sides of the membrane. That's osmosis. Now, there's also some terms that go along with osmosis, which you need to learn. The first one is called isotonic. Isotonic means equal solute concentration. So if I have the same amount of solute over here as over here, same concentration of solute on both sides of this membrane, then that is an isotonic solution. Now, if you take a cell, so this is now representing a model of a cell. So let's say this is our cell over here. We take this cell and we put it into an isotonic solution. Since there's an equal solute concentration on both sides of the membrane, there's no net direction of osmosis. In other words, in net, water is not going to be entering the cell or leaving the cell. So an example of an isotonic solution is your red blood cells. Okay, so your red blood cells are inside your bloodstream. Well, there are things dissolved inside your red blood cells. There's also stuff dissolved inside your bloodstream. Okay, the concentration of the solute inside your red blood cells is equal to the concentration of the solute inside your bloodstream, so there's no net movement of water into or out of your red blood cells. So that would be an isotonic solution, equal solute concentration. The next term is hypertonic. So this term, hypertonic, means more solute. Okay, and if you think about it, that makes sense, because hyper, you've heard that prefix before, like a kid is hyperactive is like more active, right? So hyper means more, so hypertonic means more solute. So if we take a cell and we put it into a high concentration, let's say salt solution as an example, that means we're putting that cell into a hypertonic solution. So now there is a difference in the solute concentration inside the cell compared to outside the cell. So since there's less solute inside the cell, there's more solute outside the cell, osmosis is going to happen. And the net direction of osmosis is from low solute to high solute. So what that means is water is going to leave the cell. For example, if you take your red blood cells, which should have this nice disc shape that you're seeing here, if you take your red blood cells and instead of it being in your bloodstream, you put it in a high concentration saltwater solution, what will then happen is water will start to leave your red blood cells. Your red blood cells will shrivel up, okay, because they're losing all of their water. So that's what happens if you put a cell into a hypertonic solution. Let's say we do the opposite. Instead of taking a red blood cell and putting it in a hypertonic solution, let's say we take that red blood cell and put it into a solution that has a lower concentration of solute inside the solution compared to your red blood cell. For example, let's say we take the red blood cells out of your bloodstream and put it into a beaker of like pure distilled water. Pure distilled water would have less solute outside the cell compared to inside the cell. So since you now have less solute outside the cell, okay, compared to the solute inside the cell, okay, that means there's a difference in solute concentration, so osmosis is going to happen. The direction of osmosis is from low solute concentration to high solute concentration. So what that means is water is going to enter that red blood cell, and that red blood cell is going to swell up, and then it's actually going to burst open, killing that red blood cell. Okay, so fun fact, that's why if you go to the hospital and they give you an IV, they're not injecting you with pure distilled water. Because if they were injecting you with pure distilled water, it would cause all of your red blood cells to swell up and burst open, killing your red blood cells and killing you, which would be bad for you and bad for the hospital. So instead of that, what they're doing, that IV solution is an isotonic solution, equal solute concentration compared to your red blood cells. Isotonic means equal solute concentration, and there's no net direction of osmosis. Hypertonic means higher solute concentration. If you put a cell in a hypertonic solution, water will leave the cell. Hypotonic means lower solute concentration. If you take a cell and put it in a hypotonic solution, water will enter the cell. If it's an animal cell, like one of your cells, like your red blood cells, it will swell up and burst open, killing that animal cell. Now, if it's a plant cell, the plant cell will not burst open because plant cells have a cell wall. The cell wall holds that plant cell together and prevents it from bursting open. For osmosis, the net direction of osmosis is from high water concentration to low water concentration. But remember, we don't normally say it that way. The way we would normally say is the net direction of osmosis is from low solute concentration to high solute concentration. That's the net direction of osmosis. But there's a third way you can say it at the same time. Okay, so I just told you that this is hypotonic compared to this. That's less solute over here compared to here. What that means is at the same time there's more solute over here compared to here. So what that means is technically this is hypertonic compared to this. So the net direction of osmosis is actually from hypotonic to hypertonic. Okay, so three different ways you can say it. The net direction of osmosis is from high water concentration to low water concentration. You'll basically never hear it said that way. Or it's from low solute concentration to high solute concentration. or the next direction of osmosis is from hypotonic to hypertonic. Okay, so that's osmosis. Make sure you understand what's happening to cells. You put them in these different solutions. If you take a cell and put it in an isotonic solution, there's no net direction of osmosis. If you take an animal cell and put it in a hypertonic solution, water leaves the animal cell and it shrivels up. If you take an animal cell and put it in a hypotonic solution, water enters the animal cell, it swells up and bursts open, killing that animal cell. Now, plants are similar but a little different. Okay, if you take a plant and put it in a hypotonic solution, water will start to enter that plant cell and it will start to swell up, but it won't burst open. Okay, the reason is because plants have a cell wall outside of the cell. So that cell wall holds the plant cell together so it can't burst open from too much water entering it. Now, if you put a plant in a hypertonic solution, water will leave the plant cell, the plasma membrane will pull away from the plant cell wall, and the plant cell can die from that. Alright, so this is the second out of the third video on cell membranes. Today we discussed osmosis and diffusion. In the next video, what we're going to talk about is transport, how things actually cross a membrane to get into or out of the cell. And until then, this has been Dr. Sage.